Hello students, this is continuation of tools of genetic engineering. Okay, what is the tools? Tools means the enzymes we are going to use in this genetic engineering. In the last video I explained you to cut the plasmid using a molecular scissors called restriction endonuclease enzyme. What is that? In the last video I explained you what is RE, that is a restriction endonuclease enzyme. Okay, and in the year of 1963 there is a restriction in the growth of bacteriophage in the E. coli organism. That is the growth of the bacteriophage. Bacteriophages you have studied in the last year in the lytic cycle, lysogenic cycle. Okay, bacteriophage is a virus. When it is growing in the bacteria, its growth gets restricted by the two enzymes in the year of 1963. What are the two enzymes? That is restriction endonuclease enzyme and alkaline phosphatase enzyme. Alkaline phosphatase enzyme, which is add the methyl group to the DNA. But the another enzyme, restriction endonuclease, it is cutting the DNA sequences at particular spaces, particular sites. Okay. So these two enzymes are recognized in the 1963. This restriction endonuclease enzyme or restriction, as the name indicates, the endonuclease, that is restriction. Endo means inside the nucleus, that is inside the nucleus. It's an enzyme. Always if you see the enzyme name, it is ends with the ASE. It is ends with the ASE, phosphatase, ASE. So always the enzyme name is ends with the ASE. So, okay, so this endonuclease, if you are taking the plasmid in the genetic engineering video, the recombinant DNA technology video, I had explained you restriction enzyme is a molecular sister. It is cutting the DNA. Okay, so it's cut the molecular scissor. These two type are the restriction endonuclease. One is a endo restriction nuclease, and next restriction endonuclease, exonuclease. The exonuclease is restriction endonuclease cut the DNA at the end of the DNA. The example ball 31 and exonuclease 3. If you are taking the endo restriction nuclease is in the middle of the DNA, it is cutting. Example eco R1 Hind2 BAMH1 PVU. So what is the name? How they are recognized this name? I will explain you. Okay, later I will explain you. So this enzyme, this is cutting at the middle of the DNA, this is cutting at the end of the DNA. So both this DNA is cut at the restriction site. Okay, it is going, this enzyme will read that particular place where it has to cut, it has to recognize that particular site. In that site only it cut. Okay, not the all site will not be cut by this restriction enzyme. This restriction enzyme should recognize one particular site. That particular site is called restriction site. In that particular restriction site only, this restriction endonuclease enzyme will cut. Okay. So if it is cutting, the alkaline phosphatase enzyme will add the methyl group. So it will increases the DNA strand by cutting at one place. Where it is cutting? By recognizing a restriction site they are telling. So this is the just introduction to the restriction endonuclease enzyme. In the next video I will explain you detailedly. Thank you.